Hi everyone, and thanks for listening today. In the next few minutes, I'll give a short overview of lipid nanoparticles, or LNPs, and why they've become one of the most important technologies in modern drug delivery. So most people became familiar with LNPs through the mRNA COVID-19 vaccine, but their potential extends far beyond vaccines. So one of the biggest opportunities in the field now is improving how these particles target specific tissues and cell types. I'll also touch on how VHH-based targeting strategies may help address one of the major limitations of current LNP systems. So what are lipid nanoparticles? So lipid nanoparticles are nanoscale delivery systems designed to carry therapeutic cargo such as mRNA, single-stranded RNA, or other nucleic acids. Structurally, they're composed of several lipid components that each play a different role. There are ionizable lipids that help encapsulate and release the payload, cholesterol for improved stability, helper lipids to support membrane fusion, and pegylated lipids to help control circulation time. Together, these components create a particle that can protect sensitive therapeutic cargo during circulation and support its delivery into cells. This is particularly important for RNA therapeutics because RNA molecules are extremely fragile and degrade rapidly in the body if left unprotected. LNPs therefore act as a protective barrier that shields the payload, transports it through the bloodstream, and can help it enter the target cells. Once inside the cells, the payload can then perform its therapeutic function. So LNPs have become transformative because they have solved or helped solve one of the biggest bottlenecks in nucleic acid therapeutics, delivery. So as I've mentioned on the previous slide, before these systems, RNA molecules were difficult to stabilize, difficult to deliver into cells, and rapidly degraded in circulation. So LNPs have significantly improved many of these issues. So that breakthrough enabled technologies such as mRNA vaccines and several approved RNA-based therapeutics. In many ways, the success of RNA medicine is tightly linked to the advances in nanoparticle delivery systems. So, despite their success, current lipid nanoparticle systems still have an important limitation. So most lipid nanoparticles naturally accumulate in the liver. So this happens partly because serum proteins such as ApoE bind to the particles and direct them towards the liver uptake pathways. So for liver diseases, this can actually be beneficial, but for many other applications such as oncology, neurological disorders, or immune modulation, it becomes a major obstacle. So the field is now moving towards a key question. How do we make lipid nanoparticles target the right cells and tissues more precisely? One of the major strategies being explored to improve lipid nanoparticle specificity is targeting. So the idea is relatively straightforward. By attaching a targeting moiety to the surface of the lipid nanoparticles, we may be able to direct them towards particular cell types or tissues. So these moieties can take several forms, including full-length antibodies, antibody fragments such as VHHs or CSVs, peptides, small molecules, and even aptamers that bind specific cellular receptors. So each approach has different trade-offs in terms of specificity, stability, manufacturability, and particle size. So while several targeting moieties are being explored, VHHs are generating particular interest in the nanoparticle field. So compared with full-length antibodies, VHHs are much smaller and can be easier to engineer into delivery systems without substantially increasing particle complexity. Their compact structure may also help preserve nanoparticle stability and improve manufacturability. Importantly, they can still maintain high binding specificity towards target receptors. For these reasons, many researchers see VHHs as a promising platform for enabling more selective and programmable lipid nanoparticle delivery. So if targeted delivery becomes more reliable, the implications could be significant. For example, targeted LNPs could support therapies that cross the blood-brain barrier, enable delivery into tumours, improve in vivo gene editing approaches, or enable more precise immune modulation. In other words, the future of LNPs may involve moving from passive distribution towards programmable delivery systems. That transition could substantially expand the range of diseases that RNA therapeutics can address. So, despite the excitement around targeted nanoparticles, there are several challenges we still need to be aware of. So, scientifically, achieving highly efficient and selective targeting is difficult, and endosomal escape remains relatively inefficient in many systems. From a manufacturing perspective, scalability, consistency, and particle stability are essential for commercialization and are things that need to be considered during development. And finally, there's regulation. So regulation agencies will continue to focus heavily on safety, biodistribution, and long-term effects. So these are still things that are currently being looked at. So whilst the field is progressing rapidly, there is still substantial work ahead. So I'll finish with 3 key takeaways. So first, lipid nanoparticles have enabled the modern era of RNA therapeutics, but their delivery is still biologically constrained. Second, there are 2 major bottlenecks in the system: where the particles go in the body and what happens once they enter the cell, particularly endosomal escape. And finally, the field is increasingly moving towards more programmable delivery systems. This includes ligands-based targeting approaches and emerging strategies such as VHH-based engineering, which may help improve both specificity and functionality. So overall, the focus is shifting from simply delivering RNA to delivering it to the right place and ensuring it becomes biologically active once inside the cell. Thank you for listening.